Yeah, hi everybody. This is the review for the micro exam number 2 that we're having a week from tomorrow, which would be Wednesday the 12th. And so the first part you might find it a little more difficult to listen to, depending on when you're listening to it. It's what we're going over tomorrow on Wednesday the 5th, and that is the chemical means of control. I do them by category rather than directly by name. So you're going to see categories like alcohol, aldehydes, biguanides, phenolics, halogens, uh, peroxygens, quaternary ammonium salts, things like that. So that's why, and that table, and that's the best way to classify them because there's a lot of different brand names. So, for instance, aldehydes are, and we don't do that much of it, but there's some things that you have to sterilize in liquid because they just heat just damages them tremendously. And so cold sterilization, sometimes people erroneously refer to that. So sometimes they'll talk about different kinds of radiation or pressure because effectively you're sterilizing and it's not heating, you're not using heat. So I can phrase it as cold liquid sterilization. By far and away, the agent that's used most often is aldehydes. I use them in the office. The problem is time. They typically require somewhere between 6 to 12 hours in order to be able to achieve sterilization. Uh, Very useful for heat sensitive items. So that's one of the first nuggets to remember. The other big problem is with antiseptics. There are a number of categories. I mean, alcohol is basically an antiseptic. It's not a disinfectant. I talked about that with regard to viruses. But alcohols in general, they evaporate so quickly, and that makes them useful for certain things. However, when we're looking for things that really are good antiseptics, really the two best, and they do have certain limitations. To me, the best one to utilize for surgical scrubbing for indwelling catheters, and it's something that's relatively new. They're in the category of what are called biguanides. And the name that you'll hear a lot is chlorhexidine. You'll hear the names of Hibiclens or Hexaclens. That is the go-to soap that you're going to see. You walk into UPMC at the sinks, and they're going to have these biguanides or chlorhexidine based soaps. And there's a variety of things you have dental work, you're going to get a chlorhexidine mouthwash. That's just the way it is. So it's very, very useful because it tends to adhere without irritating to mucous membranes and skin. Typically, and it's one of those that's neat because its power is enhanced when it's mixed with regular soap. And so typically how they, I don't know if they pigment it intentionally. I think so. It comes out as sort of a pink, whitish kind of a foam because the liquid portion of it is definitely pink in color. We'll be using that for my students in the lab, interestingly. The other one that used is betadine soap. And betadine is another one that when it's mixed with soap is enhanced. And those are the two things you see for surgical scrubbing, the betadine and the chlorhexidine. Betadine has a nice, distinctly brownish color. That's one of its limitations when we use betadine solution. And to me, it's very much an antiseptic. Some people, and even this textbook, will occasionally call it a disinfectant. It's really not. It's a very ineffective disinfectant. It's a very effective antiseptic. And that brownish color, you generally have to wash it off because it can obscure things. You don't want, particularly an operative site, a surgical site, to have colors like red or brown on them because it's hard to tell if something's going on underneath it post-operatively. Just saying. So we use betadine and we rinsed it off with alcohol. We didn't feel the need to leave the betadine on. One, because you're not sterilizing the skin, you're just removing surface organisms that are there. We use poison gas. I talked about that a little bit all the time. It's called ethylene oxide. It's a category all of its own. It's very effective against heat and moisture sensitive items. So we can use cold liquid for moisture, for things you can't do with heat on, when neither one of those work. And there's so many, when you see sterile disposables, the vast majority, whether they're Q-tips, whether they're Band-Aids, whether they're the packs you go buy at Walmart or Giant Eagle when you want to get a couple of sterile dressing materials or little gauze pads, those are all poison gas oriented. They put them in a big gas chamber, they seal them, the gas penetrates the packaging, and there you are. and we'll go into the nuances about it. So, I mean, it's really a very useful alternative. The gas doesn't linger whatsoever. Well, they've been around for a very long time. The halogens are probably the most used. Halogens, you remember on the periodic table, you have basically chloride, iodine, fluoride, bromide. Okay, all of those ides are basically, and it's chlorine, fluorine, bromine, it's iodine, etc. Halogens are inherently toxic to a lot of, not just us as well as microorganisms, but we utilize them. It's certainly the most common we utilize is bleach, and that's typically when we talk about Clorox, that's a chloride-based without going into the chemistry about it. That's a sterilant if you have enough time. And when we're wiping down those tables in the lab, we're diluting it. At home, you even you could even sterilize it if you had a sufficient amount of time with this. You can basically go get Clorox at wherever, at Walmart, and or even a generic Clorox, and you dilute it almost like 1 to 100. You just need 3 or 4 ounces to be roughly a gallon, and that has a great deal of activity. And if you can soak things in them, the problem is they leave a nasty residue. They're nasty to the skin. The smell isn't great. So there's a lot of limitations. We utilize that in just little sprays in the lab. It's very effective. Don't use it on stainless steel. It will tarnish it. So, but like the tabletops we have in the lab, and typically Formica type tabletops, work very, very effectively. Good product, that very inexpensive to use. Yeah, I mean, so there's a fair amount of good news with regard to that. So, and again, we'll review the nuances in lecture whenever you're listening to this. If you're listening, this will be tomorrow. If you're listening to this today, which is the 4th. And the other idea is, and it's not the only one. Think of it, our water. We fluoridate our water. And basically that, to a certain extent, that's been utilized to take care to limit the amount of cavities that form. Bromine or bromide type tablets have been used. We used to use them in our hot tub. I happen to like the Clorox products better because we have like a small hot tub, sort of sunk into our deck outside. And then, but the whole idea with iodine was very, very nasty. And we married it because it was released too quickly. It irritated the skin. It was painful. So we married it to a protein carrier called povidone. So it's released more slowly. And so the fancy name is povidone iodine, and that's what we call betadine. And it's a wonderful product. It has its limitations. It's toxic to tissues. You don't want to use it straight on wounds. I'll go into that in lecture. But it has that very distinctive brownish color, and it's enhanced by soap. And so it's one of the surgical scrubbing agents. So to be familiar with that. And then people underestimate how important metals have been over the course of time. Some of the earliest ways we treated things were with metallic products. We found that little shreds of like little bits of gold film and silver film actually limited the growth dramatically. Metals basically, if you want to know how they work, they denature the proteins directly. Metals are like that with a tremendous amount of proteins. The problem is that there's a lot of toxicity with them. So first of all, when we used to use gold and silver to fill cavities, now we use what they call non-precious metal alloys, and very often today we're even doing more cosmetic type. We're making alloys and coating them so they look more like regular teeth. So you don't have someone of my generation would have basically silver areas or gold areas in their teeth. Now they were using those metals because even the non-precious metals, because they limited the growth of bacteria. They were really very useful. We've used them a lot. The one we used, and we used it topically, was called mercurochrome or methylate. That's a mercury compound. Mercury is another one of these nasty old toxins. But as we get into in class, one of the problems with it, one is its color. And we used silver nitrate in kids' eyes, and that would leave stains on the eyes. It would stain the skin, if only temporarily. And so there have been a fair amount of uses. We're going to get into the idea of, unfortunately, with mercuric compounds, they've been using them as preservatives. Not any longer. They've gotten away from it for vaccines and for children under 6 years old. I don't think they're allowed to put them in because there was some bad and unfortunately, that was the trigger for a lot of the vaccine distrust. There was a poorly researched article that appeared in the British medical journals back in the late 90s that accused, and it's the best term, and said there was a definite link between autism and something called thimerosal, which is the mercury compound. We'll go over it in class. The research was so shoddy that this prestigious medical journal just said, "This is we've checked and checked, and now this we never should have published this." Blah 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 blah. Trouble is, the reality is they have never proven, but for that matter, they have never disproven a link between autism and mercuric compounds. Mercury is a neurotoxin. Little baby nervous systems are not mature, and you and I could tolerate that amount of mercury easily, but unfortunately, there's major questions about it. So I'll get into this all about vaccines, which is some of the stuff you need to remember. And then we utilize a wonderful product, a household product, under the right precautions that we use every day called hydrogen peroxide. It's called peroxygens. The whole idea is it releases oxygen. And a lot of bacteria can't deal with high levels of oxygen. When you force oxygen, and we use concentrated oxygen as a therapy, we'll touch on that in wounds. That's important. So remember the distinction between physical and chemical methods of control. Physical methods control heat, radiation, mechanical removal. They don't involve chemicals. They're by definition, they're physical methods of control. And remember the definitions, because there's some definitions in this. Sterilization is the removal of all microorganisms and viruses, including endospores. Again, prions are not in that, and so we don't put the language of prions either saying not prions or in prions at this point in time, because until we know specifically what's involved with it. And so collectively germicidal agents that work on surfaces and inanimate objects, disinfectants, if they work on skin, they're antiseptics. Period. Very straightforward. And again, I tried to point out to everybody that whether using Purell, and I do the experiment in the lab, that hand washing experiment, if you're doing it properly, it's all about the mechanical activity. Some things are better than others, but not so much as you would find statistically significantly. So, I mean, in the absence of soap of any type, just plain water works. Really, I mean, sometimes you don't have, depending on the circumstances, you don't have the luxury of doing certain things. So you use what's available, and you'd be surprised how well it works when we do that. Remember the sterilization number, 121 degrees Celsius for 15 minutes. Okay, and basically that generates 15 pounds per square inch. That's the autoclave number for sterilization. And again, the fact that it turns autoclave tape black, or chemical indicators change a particular color, and we do use a lot of those, that is a chemical test. Sterilization is a biological test. You have to have an endospore containing tube that you autoclave, and then you attempt to grow it in culture afterwards. That's the way that when you're dealing with documentation and you're dealing with hospital-based things, you can't be fast and loose with the rules. It's basically another way of what we would call standardizing. It would be the equivalent would be if you're doing blood. Look, if you have a glucometer at home and someone's a diabetic and they keep getting a blood sugar of 95, they may be very happy, but maybe you should check to make sure it's not broken, and just every time you use it, it reaches 95. That's the idea of calibrating, and effectively you're just ascertaining that the autoclave works properly. That's where that pack of stuff goes in, and why it's so appropriate. So those are important. Remember the numbers for pasteurization. The batch method is the one that takes 30 minutes and roughly 62 to 63 degrees Celsius. That's about 140 degrees Fahrenheit. The HTST, or high temperature short time, pumps up another 10 degrees Celsius, roughly 18 degrees. If you want to think of it that way, that pumps it up another 18 degrees Fahrenheit, and then you can reduce this to roughly 15 or 20 seconds. If it has a lot of fats, we pump it up yet another 10 degrees Celsius, and maybe add 20 to 30 seconds. Those are much more commercial usage of pasteurization. And remember, the difference in commercial sterilization is that the target organism or the target endospore is botulism, because all the other endospores are not basic, are not effective byways of consumables. Those endospores don't harm you. The botulism can be a big time player. Okay. So these are all different sorts of things that you have to recall. Hydrogen peroxide is so versatile because it can be an antiseptic or a disinfectant, and indeed it can be a sterilant. When we start talking about, I mentioned that ultra pasteurization, I go over it again, really is a type of sterilization. We use what's called vapor phase hydrogen peroxide, superheated, and evaporates so quickly, and that's the beauty of it, and that's why it's sort of eco-friendly, and that's another thing to consider, because a lot of these chemicals are just nasty. You smell them, you can't breathe them, you couldn't use it with somebody with respiratory problems. So why would you use that in a room to clean the floor or the countertops when you have a patient in there? You wouldn't. So you go more to eco-friendly things, and hydrogen peroxide has helped those become much more effective. The vapor phase evaporates so quickly, and it sterilizes the inside of these containers like juice boxes. And they have a very interesting sort of a sandwich layer construction that allows them to be shelf stable for long periods of time, as long as they're not open. And again, the big deal about alcohol, good news and bad news, same thing, they evaporate quickly. Great for those hand sanitizers, they evaporate quickly. You're not dripping water on the floor. Bad for hand sanitizers because they evaporate quickly and they don't leave a residue. So it's good news and bad news. There's nothing perfect. I mentioned in passing what are called quaternary ammonium salts. Quats is a nickname. That's the ingredient that we utilize today in antibacterial soaps. And you see, years ago there have been other ingredients. They've all had certain degrees of toxicity. They were mostly in the phenolic area. So phenols are still used. That was the original stuff that Lister used. But unfortunately, phenols have had their problems, and attempts to use phenolic compounds, although they've been worked for a while, they started to see the potential of being absorbed systemically, and you didn't want to open that to problems. So these quaternary ammonium salts, the good news, they're very useful on your skin. Again, the mechanical action is really the trigger. This helps. Is it better than hand soap? I don't think so. Is it better than some of the surgical scrubbing things that we use? I don't think so. But is it still useful for everyday usage? For sure. But understand, this is the same stuff that they're wiping down tables with in a restaurant. They're the same stuff, and you will see this in the PowerPoint that is utilized when you see Clorox wipes and Lysol wipes. They don't have Clorox or Lysol. No, that stuff is unpleasant to skin directly, and sometimes has a funky odor. They use quaternary ammonium salts. They've been around for a while. We'll go over them as well. Now, the bulk of the questions, though, I think, are more about viruses. So you have to understand how we classify them. Remember genomes, envelopes, their architecture or shape. The ones either isometric or icosahedral, they're the same thing. Filamentous or helical, pleiform or complex. We went over this. Viruses are very versatile. If it's a living thing, viruses can infect it. You got to remember that. And then look at the ones, again, I'm interested in basically have an idea of what condition we spent a bunch of time, and it's the first one we looked at for HPV. Basically, they cause warts, all sorts of warts, including genital warts. The problem is they're a naked virus. The isometric really doesn't matter, but because that's the most common type, but they're a naked virus. They're a DNA virus, which means once you get them, because they can become latent, you have them effectively forever. It doesn't mean they're going to show up again, but the potential, depending on this particular type or strain of virus, and we call them types, could lead to some type of potential genital warts in either the male or the female that could certainly become cancerous. Big player. Like I said, 15 to 30% of all cancers are directly related to viral infection. And in my opinion, more than half of those are all about HPV. You want to stay away from it if you possibly can. Not necessarily so easy. I mean, we go into warts a couple of times and really probably spend more time about some of the problems that are attendant to it when we get into urinary tract infections. Do have some more specifics. Pardon me. Coffee break. Remember, the shape is based on the capsid. That's the protein enclosure. It's made of capsomeres. In a non-enveloped virus, those protein spikes protrude from the capsid. In the enveloped one, the spikes protrude from the envelope itself, not from the capsid. That makes the envelope a great target for disinfectants and antiseptics. That's why it's much easier to control enveloped viruses like HIV, like hepatitis C, like coronaviruses, as opposed to the common cold, which is a rhinovirus, or HPV, which we just talked about. So the protein spikes are how they attach to specific receptors. That's really important. The one we see the most is that icosahedral or isometric virus, a capsid design rather. That it's made of the capsomeres, and it's roughly 20 of them. They form it. It's a 20-sided figure, okay, and basically they're equilateral triangles. That's the way to describe it. And so it's like a 20-sided die. That's so very famous, either icosahedral. Most of the time we use isometric for same side. And again, to paint with a broad brush, DNA viruses frequently become latent, meaning the DNA incorporates with the host DNA and can alter it either transiently or permanently. Okay, and unfortunately, there's along with HPV, herpes viruses like the Kaposi sarcoma or Epstein-Barr virus have certainly been associated with cancers. So there's some significant problems associated with them. And the idea of latency, true for HSV1, HSV2, that's respectively oral and genital herpes, even though they could interchange. And again, we review a lot of those again when we look at we focus more on the diseases, particularly the urinary tract and sexually transmitted diseases that are there. We focus on VSV, varicella zoster virus, chickenpox to shingles, and that's notoriously latent. And shingles is not a picnic at all. It can be very, very problematic. Nobody wants to get it. Okay, for instance, and then you should know the categories in general, like picoRNA viruses, the common cold, enterovirus. You have rhinoviruses, enteroviruses, polio's in there, hepatitis A is in there. They're all in that category. They're a small, naked, basically isometric virus, and that's why they trouble us so much. They're always there. You can see that, and because it affects all of us, and unfortunately with a certain degree of frequency. So that's another number. So you should know nuggets of those little categories, like a Rio virus was that rotavirus, the infantile diarrhea virus. HIV, okay, is a retrovirus. Okay, parvovirus, even though it affects mostly animals, it's that one that slap cheek syndrome, and so it can affect us in skin. And we're going to talk about how they spread, etc., etc. So that's part of it. So one of the questions vis-a-vis how they spread, here's the answer key: A, B, C, and D in this order: fecal to oral, respiratory, zoonotic, sexually transmitted, or blood or body fluids. And I have like 5 diseases we talked about. They're on the study guide. Figure out which one is, and they're not all the same. Okay, so it's one of those use once, twice, or not at all. Typically, the way I do it. Understand what goes on when viruses infect us. They have to first they have to attach, then they have to uncoat. Then once they uncoat it, you get basically inside one of our cells. The capsid goes away, and now the genetic material effectively takes over and hijacks the cell mechanism. Again, if it's DNA, it'll start making proteins. Even if it's RNA, basically, it starts making protein. The problem with RNA viruses is inherently the reproduction of RNA has a lot more potential for mutations. And indeed, some of these ones we talked a little bit about, but it's not a focus point on antigenic shift and drift until we really get into respiratory viruses, has to do with RNA viruses. So whether it's RSV, whether it's influenza, whether it's coronaviruses, the inherent problem with them is even though DNA viruses undergo a certain amount of mutation or genetic variation, it's not nearly as frequent. not nearly as frequent. And remember, viruses basically, it's like a little assembly line. They don't do mitosis, they don't do meiosis, they don't do binary fission. They have an odd way, unique is a better way. I don't know of anything else that basically reproduces its component parts and then assembles them. Okay. So understand where the categories are. Remember, and I have one of these questions with categories: orthomyxo, influenza, paramyxo, measles, mumps, RSV, something called parainfluenza, sort of a milder influenza type problem. Coronaviruses, obviously COVID-19, SARS, which actually SARS-CoV-2 is the actual scientific name for COVID-19. That's just sort of the nickname we gave it for convenience. And something called MERS, MERS, that Middle East Respiratory Syndrome. They're all in the coronavirus family. So there's a lot of those, and I have tried to put them all on there. And remember, some viruses, lytic viruses, typically the naked viruses, once they get in one of our cells, they grow and grow, and then they burst. And the cell literally bursts open. They secrete enzymes to create apoptosis. So the cell effectively will die. In this case, it bursts and it releases a large number of these. So if a burst size is 500, you start with one virus, it infects one cell, it bursts and releases 500 identical viruses, all nasty. They affect 500 cells, then the whole thing happens again, and now those 500 can each affect 500 different cells, and you get the idea how rapidly viruses can overcome you. But in the envelope ones, typically it's called budding or shedding. It's almost like the cell stays intact, but it pops them out. But remember, in either case, when a virus infects your cell, the cell is never going to get better. You might get better from the cold. Those cells are going to die. They're either going to die naturally because they have a lifespan, or they're going to die because they exploded, but they're never going to get better. When it becomes a chronic or persistent virus, a lot of times those cells, and that's really what happens in HIV, the most important white blood cell you have in your body, the helper T cell, now becomes a little factory that constantly makes viruses, infects other helper T cells, and they all start to do the same thing. And so they no longer are functional helper T cells. And that's really when we get to the true definition of AIDS. When your helper T cell count of functional, useful, working helper T cells falls below a certain number, at that point your immune system is collapsed and you're going to die sooner than later. That's what fatal is. Viruses do their damage by impacting cells. They might destroy cells. They might cause cells to fuse. They may incorporate with the host cell's DNA and again turn them into a factory. Well, they don't make toxins like bacteria. They don't cause endotoxin or exotoxic problems. It's a different mechanism. It can be just as bad. So the rash that you see, typically, when viruses, respiratory viruses particularly, get fever and a rash, that has to do with the immunological response, the fever, and the rash, probably because tiny blood vessels under the skin, the lining cells, which we call endothelium, which we're learning about now in A&P, are compromised, and you get bleeding into those areas. So I think all of those are important. So be familiar with all the viruses that affect us that we went over in class, and ignore the ones we missed. Okay, I think that's really an important aspect of it. Again, big categories to know about. Those flaviviruses and they're pretty much all carried by mosquitoes except for hepatitis C, which is obviously sexually transmitted, but that's yellow fever. That flava or flavus implies yellow. Dengue, which is something you see down in, I beg your pardon, the dengue, which is something that you see in South America, again associated with mosquitoes, something you're a little concerned about when you travel into those areas of mosquito. Anything is mosquito prone. That Zika virus you heard so much about, West Nile virus, those are all in that particular. So it's a pretty interesting category that's there. And so again, know those. I've covered all of these ones that you should be familiar with. The category. I have another one of these where I'll give you the categories that they're in, like herpes viruses or orthomyxo or paramyxo, and you need to know the viruses we talked about in those categories. And very lastly, I'm going to have a brief word about mutations. We covered a little bit of it in radiation. Ultraviolet causes something I put on the board called thymine dimers. The most powerful of all the mutagens, the things that can cause mutations and cause damage, and why we use it a lot is that radiation aspect of the thing. And those are gamma rays or X rays. Collectively, they're called ionizing radiation. They create ions, those are the charged particles. That's what does the damage to DNA directly. Where thymine dimers, they just alter the DNA. They don't necessarily destroy it or fragment it. They make those thymine dimers, which means it can't unzip and unzip. So that's called non ionizing radiation. And so that's really important. Again, what is a mutation? Anything, whether it's a chemical or physical thing like radiation or certain chemicals, that can cause an error in DNA. That's a mutation. It happens all the time when our cells replicate. It happens when bacterial cells replicate, but more frequently. We call those spontaneous mutations. The difference, so that's normal circumstances. Our cells probably mutate 1 in 1 billion times. Bacteria, maybe 1,000 times more frequently. When we add a mutagen, like we apply ionizing radiation, we apply some type of noxious chemical. That is, we call that an induced mutation. For instance, when you expose yourself to excessive amounts of alcohol, or you smoke, or you do some kind of toxin gets in your system inadvertently or by intent, okay, at that point you're inducing or causing more mutations to form. And lastly, the last thing I'll tell you about, and we're going to do that in class, probably I would say at the end of this, I'll try to make a mental note. It's called the Ames test. It's one of the things in genetics that you need to be familiar with. I'm not going to do a whole lot of genetics to test you on. I'll probably mention it either certainly before the exam on next Wednesday. It's called the Ames test. It involves salmonella, and it involves what's called a backwards or reverse mutation. And it has to be part of this test because that's something that you have to know. Everything has to go everything that the FDA is going to approve, whether it's a consumable, whether it's a medication, has to pass the Ames test, which is a very reliable way on determining whether or not something is liable to cause cancer in us. And I'll leave it at that. That concludes this presentation. I will send it to you forthwith whenever it comes through, and I'll send it to you by way of the YouTube. And thank you.